So if we want to look at the synonymous and non-synonymous substitution rate, first you have to be able to recognize. And second, as we look at nucleotide substitutions, now we also want to estimate the substitution rate for these two types of changes. Of course, it's a little bit complicated than just look at the A, T, and GC, right? Because now we have to look at the DNA sequence, and we have to know the codons, and also we have to know how each codon translated to which amino acid, and those are the important essential part. And I don't really want you to memorize the codon table. So I think practically when you look at your own sequences, the computer usually does all the work for you. But you also can have a codon table in hand. At least you have an idea the differences you are looking at. And in the class, we'll have an exam in 2 weeks, right? After the spring break and another week, we'll have an exam. I will provide you the codon table, so you will be able to calculate or at least count the synonymous and non-synonymous changes. And in your entire life, you have to count it once in our class. It's a little bit complicated, but it will take you a lot of time. So then you will appreciate how your computer works so hard for you. But anyway, just let you know how we do that. And we'll explain it a little bit at today's class. Okay, so we usually have two sets of symbols you will see from your textbook, from like the papers. So some people use DS or DN for synonymous changes and non-synonymous changes. And some people use KS or KN for synonymous and non-synonymous changes. And I don't think we are trying to unify them, but they are like different symbols for these two types of changes. And they are different methods, but it's not that complicated once you understand how we try to do and what we try to do. So basically they are counting methods or likelihood estimates. Okay, so we want to start with the simple counting method. So counting method, by definition, you know, just count. That's why in your entire life you have to count it once. So you have to count each side. It's a difficult part. You're not only going to look at each changes. You have to look at each side to decide whether it's a synonymous or non-synonymous side. And we'll talk about in a couple of slides later. Then you have to count the differences. And the difference is whether this change is a synonymous or a non-synonymous. This part is relatively easy. And once you have these two numbers, you will calculate as the nucleotide substitution. You could calculate the proportion of changes based on 1 and 2. Then we can do correction, right? So the correction, just as I said, you use different models to correct for multiple hits. Okay, here is the codon table. Just remind you what we say from the top, like angle that I'm showing here, the UUU is a TTT, I said in previous slides, and code for phenylalanine. Then at the bottom is like a GGG, so you can see like our CCC is equally distant, right? I have to require multiple changes to get down to that codon. So that's what we mean in a couple slides later, the distance from one amino acid change to another amino acid, just by counting how many mutations required for this type of changes. And they are quite different. Some only 1, 2, or 3. And you have to further consider is a transition or transversion. So that's something we can estimate or can count and can think about how that contributes to like the protein sequence differences we observed in natural population. Okay, so here is the codon table. Then they present each letter at a different direction, but basically just arranged into blocks. For each block, usually there are 2 or 1 amino acid, right? As showing on the top one. So it's either phenylalanine or leucine. So that's 2 amino acid in that block. And for block next to it is serine. So it's UCU, but any changes at the 3rd position doesn't affect the amino acid encoded to. So that's the serine. So you can look at the codon table, and I just try to remind you the possible changes at the other corner, so you know, like in your mind, there's a model at nucleotide level. So Ney and Gojobori is the first one try to estimate the different substitution rate, the synonymous and the non-synonymous, or I should say, a lot of people use this method nowadays just by the simple counting method. And this is try to show you how they count the differences and also how to estimate. And here is very simple. We use the Duke and Canton's model. That's one parent model to calculate. And you'll see this type of notation quite a lot. I just want to say one word here. It's like a JC69. And when you use a program like Mega or some others, they will do the same thing. They use the letters for the authors, so the initial for the authors like Jukes and Cantos use J and C, and the number next to it is the year they publish. So most of the work was published in 19 something, so in last century. So you see, like I said, this Jukes and Cantos and published in 1969, and they use that as a model in the computer program. I have seen quite a few computer programs use the same notation. So you also see people put like NG, or they just use NA and 86, something like that. Okay, just try to show you all the possible changes. So I say it's not too difficult to count the differences, because you just spot the differences and try to see the position within that particular codon. Then look at that particular codon at the codon table and decide whether it's a synonymous or non-synonymous. So this is relatively easy. But for any given codon, for every site, you have to look at whether it's a transition transversion. It's a lot of work. So I just want to show you one such example. So I use color code here. To start with, I use T instead of U from the previous codon table. But I start with the codon called TTT. And there are 3 positions. I use color for each position. So on the left side of this slide, I change the first position to CTT, ATT, and GTT. And if I copy correctly from the codon table, then that's the amino acid for each codon. Now, you look at the first letter of the first position of this codon T, then can you tell me whether the first position is a synonymous site or non-synonymous site? I hear one voice here, non-synonymous. Do everybody agree? Yes, right? So that's the difficult one. There's no change, right? This is just this position. So you have to think about what are the possibilities. All the possibilities are here, right? All the possibilities, instead of no change, all the possibilities are here. They all, so for any change at this position, they change the amino acid sequences. So in this case, this one is obvious, non-synonymous site. Okay, so let's look at the second position. So the second position also, I put up and down. Actually, I can put on the same same places, but anyway, this is how I arranged it. So if they can change the same thing, G, C, and T. And similar to the first one, once they change to a different amino acid. And this is, we all know from our biology. The second position, every time the second position change, they change the amino acid. So for second position, you don't really need a codon table. If you found the exception, please let me know. For second position, you can just like close your eye, just write it's a non-synonymous site, right? Okay, so this is easy. And the third position, now the third position is on the right-hand side of the slide. Okay, so now we look at, it's the same thing from T to C to A to G. Actually, that's the first column we just look at on the codon table. So if we change to C, it's phenylalanine. If we change to A or G, it's leucine. So now the question is the third position of this codon a synonymous site or a non-synonymous site? Any answers? Both? I heard both. How do we count it? Because we have to have a number. What would be the number we put on this site? So I already heard that the answer is both. Okay, what's the number for synonymous or non-synonymous site? Okay, let me put it here. For the first position, so we want to do that from codon to codon, right? So we want to calculate number of synonymous sites, okay, equal to the first position and number of non-synonymous sites. Okay, so the first and second is 1. So non-synonymous for the first position is 1. And non-synonymous for the second position is 1. If you want me to make more clear, it's 0 for the first one. It's 0 for the second one. Okay, so can someone tell me for the third position, what's the number I should put in here? Great. Okay, are this correct? Okay, let's look at the slide here. It's a phenylalanine. If T changed to C, based on the codon table, it still encodes phenylalanine, right? If the T changed to A or G, the amino acid change from phenylalanine to leucine. So from phenylalanine to leucine, that's the non-synonymous. From phenylalanine to phenylalanine, it's a synonymous. Right. So for the third position, they are 1/3 of the time, it is a synonymous site. And 2/3 of the time, it is a non-synonymous site. Okay, so that's why I say it's difficult. It's time consuming because you have to look at each individual one. And also, there are possibilities at a different time. So for the third position here, like 1/3 of the time, it's a synonymous. 2/3 of the time, it's a non-synonymous. So it's not 0 or 1 all the time. So that's why I want to put it here. So 1/3 of the time, it's a synonymous. 2/3 of the time, it's a non-synonymous. And you have to do it for every codon to calculate the number of sites in each class. So this is no differences. You haven't spotted any differences yet. It's just look at the sequence and decide whether it's a synonymous or a non-synonymous site. So you have to do that for every site. And this is just to show you what might occur in your test. Okay, I'll give you short sequences and ask you the number of synonymous sites and the number of non-synonymous sites as just shown here. And the second thing you need to do is to spot the changes. Okay, here comes two sequences. Then you have to, like here I boxed the changes along the sequence. And you have to look at the codon table and write the amino acid. And this is the differences. So the differences are either synonymous differences or non-synonymous differences. So that's the second thing you need to do. Then you calculate the proportion of synonymous changes and non-synonymous changes. So that's the counting method or anything we start with. The second part, now we have to also think about the possible changes happening or the evolutionary path. Just give you two examples. If a codon, so in the previous slide, this is a very simple version. For each codon, I only have one differences. So that's very simple. But if you have two changes on the same codon, as shown in this slide, now the question is how the two mutations or the minimal two mutations occur in sequence. So whether the first, the middle, the second position C changed to A occurred before the third position changed to A, or the third position changed to A took place earlier than the second position changes. So this is something we have to think about the possibility. Of course, we don't know. If just give it the information here, nobody would know what happened in the past. So we have to estimate all possibilities, or just list all the possibilities. So there's a past one and past two. It's very simple. Just try to say, okay, the first change is at the second position, or the first change is at the third position. So that's just two possibilities. But you have to list the changes here and decide it's a synonymous or a non-synonymous. I use replacement here. Like you have to decide based on the codon table whether this mutation changed the amino acid sequence. Then you have to say, okay, what should I write down for the differences, right? So this is something after this one, we have to write or calculate the difference in changes. Now you have two possibilities. The first one, there's a synonymous and the non-synonymous. The second one, both are non-synonymous changes. Then you say, okay, I cannot decide. Both are equally possible. Then you say, okay, half of the chance past one happened. The other half the chance past two happened. That's an equal weight estimate. So I don't decide. I'm very conservative. I don't know which one is more possible than the other one. So I say each one has equal possible rate of happening. So half of the time could be past one. The other half of time could be past two. So I time 0.5 for each possibility. The other one could look at the sequence and say, well, it's probably more likely the synonymous site is more likely to be maintained in the population than the non-synonymous site. And if your chemistry is very good, you can look at the amino acid and think about empirically how likely, or based on their chemical property, how likely this type of change could happen in the past. And you say, okay, let's give a different weight. So maybe one would decide the past one is more likely to occur evolutionarily than the second pass. So we'll say, okay, let's give past one 0.7, then pass 2, 0.3. They add up is 1, right? Then that's like a weighting method. I weight one of the possibility heavier than the other one. So I have a weighting between the two possibilities. Either way is okay. Yeah, so you have to know what you are doing, and that's the most important part. But so if a codon has 2 changes, and that's something in addition to synonymous, non-synonymous, there's something you have to decide how you want to estimate the different changes occurred during evolutionary time. Okay, so now you all know how to count the number, count the differences, then the rest is very easy. So you will be able to calculate the 2p for me, little p for me. So the little p is the proportion of the differences for synonymous or for non-synonymous. So that's the one showing on the top. Then you could decide which correction you want to do. You can use 1 parameter or 2 parameter method to calculate, to estimate the substitution rate. So this is basically the counting method.